today we are going to focus on reproducibility in the field of brain imaging. So I'm a neuroinformatician, so this is my field of research. And sorry, can you still see my screen? I just closed my laptop just to check.
So we can still see the screen, but still the first, slide.
The second one, is it?
Yes, it's good.
Okay, great. So everything is working, so I can get going now. Thank you. So what I often say about reproducible research that I really like is that often when you chat with people, a lot of people relate to this field of research. But when chatting with different people, I often realize that reproducibility means different things to different people. So by way of an introduction, I just wanted to set the terms that are going to be used in the rest of this presentation. But as I've said earlier, so I'm a researcher in brain imaging. So the type of data I'm working with is what we can see here. So here on the left, you have a couple of brain images. Then we're going to perform some analysis on this data. And our results, again, are other brain images. But in practice, when I chat about reproducibility with people more in neuroscience or neuroimaging, people who are interested about how the brain works, basically what they focus on, so you can see here on the left, I've described the original study with this circle for data and then the arrow for analysis. It's something like this. So basically, when we have a result that we identify as part of a paper, what we really want to say is, okay, what I found about the brain in this experiment, I'd like this to remain true if I work with a data set that is slightly different and using an analysis that is slightly different. That's what people in neuroscience, neuroimaging, and applicative fields in general are mostly interested in. And of course, we all know some of those findings that are super robust, strong effect. And when we try again doing roughly the same protocol, well, it reproduced well. So a good example is in neuroimaging. If you move your hand, basically you're going to have an area at the top of the brain that activates. And this is very strong and very, very reproducible across different people. Now, that being said, we've probably all made the experience that sometimes, well, it doesn't work so well and our results do not reproduce. However we are trying, we cannot get the same results as the one that were published in the original study. And there we face a lot of questions. Okay, we can't reproduce the results, but is this because I actually did not understand correctly the method as it was described in the paper? Or maybe the method is fine, but it's because I use a different data set. Or maybe data set is okay, but my computing platform has some weird effect on how the analysis behave. Or maybe sometimes it's more related to details that were actually not in the paper. So maybe, SPM is a very famous software in neuroimaging. Maybe the specific version of the software wasn't shared, and maybe I used a different version, and that explains my differences. Or sometimes some algorithms we don't describe in details, and I don't know the details of the registration algorithm, and so on and so forth. And basically, this has led, as you know, for people in the audience of this seminar series, to a lot of work on reproducibility, because obviously we want to understand what's going on here. And so in the following, I'm going to mention to create the base to what I'm going to discuss in the following. I'm going to look at different types of reproduction. So what I've just described is what people immediately think about when I chat with neuroimagers. Now, when I chat with computer scientists, because as a neuroinformatician, I'm in between the two communities, often they think about the reproduction that we see at the top left here. So basically the idea that we want to be able to reobtain the very same results under the very same conditions, same data, same analysis. And basically, those two extremes, so I'm going to call in the following computational reproducibility and replicability, Of course, this is a choice of mine. There are a number of definitions out there, and sometimes they are contradicting, but that's what I'm going to base my talk on. And what we have here, basically, we could build a matrix where we have on one axis different data, same data, and on the other axis, same analysis, different analysis. And of course, I have only 4 different subtypes of reproduction here, but I could have many more because different versus same data could be done in many different ways. Maybe I have another sample of the same population. Maybe I'm looking at another population, people in a different geographical location, for instance. Maybe I'm looking at young kids versus adults, and so on and so forth. And what's quite interesting is that depending on where we're standing in that matrix, a lack of reproducibility will not call for the same solutions. So, for instance, when we think about computational reproducibility, the idea that we'd like to have the ability, if we have the same data and the same analysis, to reconstruct the same results, there are a lot of questions around transparency, so sharing the code that produced the analysis, around data sharing, and this can be particularly complex in the field of biomedical research with human participants, because we have basically to comply with different regulation, including the GDPR for us. So this is computational reproducibility, but beyond the question of transparency and having access to the actual artifacts, there are also questions about how can we rerun the experiment with the very same environment, and how can we actually share enough details about this environment, including the libraries that were used, but also the system and all the low-level libraries. So that's one aspect of reproducibility. Another aspect that has been looked a lot, I would say, in brain imaging, and that's probably what the first studies were looking at, is the idea to try and rerun the same analysis, but with a different data set. And this led in our community to a lot of reflection on the size of the data set that we use, the statistical power to detect an effect or not, and has led to a lot of efforts toward more data sharing. Today, I'm mainly gonna focus on this top right-hand corner type of reproducibility. So basically, we start with the same data, but we're going to vary the analysis, and the idea is to understand the impact it has on our results. And this can be seen as a robustness test of our results. And of course, I'll get into way more details in the following. So for a long time in our community, people were, when we were chatting about reproducibility and saying that we could use different analysis, there was this idea that basically different analysis, because they were built with the expertise of the neuroimager, should not really impact the final results. But only recently this has been actually tested, and I'll present the results just now. But before I do that, I want to give you a better idea of what's an analysis in brain imaging, and why do we think we might have multiple ways to analyze the same data set. So I'm going to take my small schematic that I had earlier, zoom in a little bit more. So again, the input data, the analysis pipeline, and the results. But before I get into the details of how a pipeline looks like, I just want to say a few words about the type of data we're working with. So as input data in my research, I work with brain imaging from human participants, mainly coming from magnetic resonance imaging. So you have on this picture at the bottom, the MR scan. Basically, the participant is going to lie in the machine, and their brain is going to get imaged. So that's the type of data we get as an input. As an output, we get activation maps. So this is a specific type of brain imaging where we're looking at functional MRI. So here is an example of activation maps. So how this looks like is that In the background, we have a picture of the brain. That's what you see in gray levels. And then on top of that, the overlay is going to be the regions that are activated while the person was doing a specific task in the scanner. And task can be a lot of different things depending on the experiment. So here, for instance, we have the activations, the brain areas that are activated when listening to music. But we could also look at the brain areas that are activating when we are looking at pictures, for instance, or the brain areas that are activated when I move my hand, and so on and so forth. And basically, neuroscientists can vary the type of tasks they are asking the participant to do while in the machine. And in the end, they get these activation maps that they can study. So now, going back to our analysis pipeline. So basically, the idea is that we want to transform our input data, which is basically brain pictures that are taken, so 3D images, one after the other, while the person in the scanner is doing the task of interest. And we want to analyze these images so that we can get our results, which are the locations in the brain that are activated while doing the task that was defined in the protocol. But in practice, we can typically resplit that pipeline into 2 high-level steps. And this is true not only in brain imaging, but in general in research with data, I would say. So basically, typically you have a phase of data preparation. You're going to denoise your image. So we have, for instance, inhomogeneities in terms of intensities. We're going to correct for that. But data preparation can also include, for us, normalization. So we need to put the brains of all the different people in the same standardized space so that we can compare them. So that's the first step. Then we get our derived data. So that's our measure of interest. And then we can do a statistical analysis to test what we are interested in. But in practice, of course, those 2 high-level steps, it's not as simple as that. It's more something like this, and could be even more complex as a tree, where we have steps that happen one after the other. So these steps could include motion correction. I was talking earlier of registration to a standardized space, segmentation, and so on and so forth, up to the final statistical analysis. And those are the steps of my pipeline. But in practice, for each of the steps that I can see in this pipeline, as a practitioner, I'm going to face a number of choices. So first, often, there are a number of algorithms that I could have used for that particular step. If I stick to a given algorithm, because, well, I have previous knowledge that this is the best algorithm for my particular use case, then often those are implemented in different software packages. Even if I know the software package that I want to use, they will be typically available in different software versions. Even if I stick to a given version, there are a number of parameters that may vary and may be considered as acceptable for my use case. And even if I fix the parameters, I could also run these particular analysis in a different software environment. And all of this taken together creates not only a single pipeline that makes it possible to go from my input data to my final results, but rather a family of acceptable pipelines. So I've put 4 examples here that could be considered as something that would typically happen. So the second one is the same pipeline, but with an earlier version of the software. And as it turns out, then I have to change the parameters of the third step because in that version, that's how it worked. The third version of the pipeline is the same, but using a different software. And as it turns out, for steps 2 and 4, in that software, the algorithm that is used is different. And the last one is the same pipeline, but changing the parameters of the pre-processing steps. And it's really easy to see that if we mix and match all of these options very quickly, it becomes like a huge number of combinations. And so, as I was saying earlier, for quite some time, we were considering in the community that although there were a number of combinations, these should not impact our final results. And then there was this many analyst project called NAPS. So many analyst projects are those projects that have been going on in different scientific communities, where basically we have on one hand one data set, and then a number of research questions. So here there were 9, and researchers had to answer using the same data set, those 9 questions that could be answered with yes or no. So just to give a sense of the type of questions, this is what we had for question 1. So the question was, is there a parametric effect of gain positive in the prefrontal ventromedial cortex for the indifference group? So if I try to simplify this a little bit, so basically the task here was a gambling task. So parametric effect of gain is looking at activations that would be growing in a proportional way to the gain, the money gain that the people could get in the experiment. The prefrontal ventromedial cortex is just one of the brain region, and the indifference group is a subgroup of the participants that had done a slightly different version of the task. So basically we have a number of participants and we are looking at activity in their brain, and if there is an activity in a given predefined region, we answer yes. If there is no, we answer no. And the project worked like this. They recruited using social media and different mailing list teams, analysis teams that worked completely independently. And in the end, they did manage to recruit 70 teams. So these teams were like 1, 2, 3 people max. And they had to answer the question using the data set, and they could use the pipeline that they believe would be the most suitable, or maybe their favorite pipeline to answer the research questions. And the results were as follows. So to read this graph, basically you have on the x-axis the 9 yes or no research questions. And then the red dot corresponds to the proportion of teams that replied yes or no. So basically, when we are at the top 80% mean that 80% of the teams agree to answer a yes to the actual question. So that's the case for question hypothesis 5 here on the right. And at the bottom, 80% of the teams agree to answer no to the research question. So the results are non-significant. And here we have 3 hypotheses, 7, 8, and 9, where the team agreed to answer no. But then we have the bulk of hypotheses that are in the middle. And basically here, we have contradicting results across the teams. So if we take the worst hypothesis, which is hypothesis 1, we have roughly a 40-60 split. And of course, this created a lot of questioning in the community, because it was unexpected that we could get only varying the pipeline, starting from the very same data and looking at a very concrete and well-defined objective, different results across scientific teams. And this has called a lot of research into trying to understand what's going on. Why are we getting different results when we have different pipelines? And I would say, and I think this is challenging. This is challenging because there might be different reasons, again, that explain this variability we observe in the result. On the one hand, we have variability that is really unwanted and would like to get rid of. So there are a number of work, for instance, that look at numerical uncertainty. If I use a different operating system and I'm getting different results for my scientific study, probably it's telling me something about the validity of my results. If such a small change implies a change in my scientific findings, then probably I should question the method in between. So that's unwanted variability. I could put in there different implementation of the same algorithm. Probably we wouldn't want those to lead to different results. Then we have some variability that is an interaction with what's happening in the data. Sometimes I'm going to get different results as of different pipeline, not due to my pipelines themselves, but maybe because the data I had was not suitable to answer my research question. Maybe I had not enough statistical power, and that means results that are very unreliable. And this unreliability we can see we can pull by pulling different pipelines. Sometimes some of my pipelines will violate the assumptions of my data. And again, that's an interaction in the data. And then finally, and I think this is why there is so much discussion around analytical variability, so the variability induced by different pipelines, we have this variability of interest. Why do we have different algorithms? Why do we have different ways to do the statistical analysis? Because basically we want ways to study the phenomenon we're looking at. And sometimes the results of different pipelines giving me different answers is going to inform me on my scientific problem, on my scientific question. And I really want to keep that variability and to expose it to scientists. And so when we work with analytical variability, we have this full spectrum, and depending on where we're standing, again, the solutions are going to be different. In the following, I'm going to do a short focus on the variability across neuroimaging software. John work that was done in collaboration with Alex Bowring and Tom Nichols from Oxford University. And so basically here we are again with our pipeline. We have some data, we have our results. And the idea is that we wanted to look specifically at the variability that observed in the fMRI results when using a different software package, a different neuroimaging software package. So what we had with NARPS is like complete flexibility where different teams would use the tools they would use in real life. Here we wanted instead to look specifically at the impact of different neuroimaging software packages. So what we've done here is that we took 3 studies from the literatures that were selected because for a number of properties, but in particular because they shared the data associated with the studies that we could actually reproduce. And we used 3 fMRI software, AFNI, FSL, and SPM. So the numbers are a bit old now, but those are 3 of the main open source software, and they cover a good range of the actual task fMRI literature. And then our work was to build pipelines that were as aligned as possible to the original pipeline used in the original paper, using what was made available to practitioners in the different software packages. And the results are as follows. So here, each column, so we have 3 columns that correspond to the 3 studies that we reproduce, and each line is a different software package, and the line at the bottom is the figure from the original paper. So first, you can observe that the figure in the original paper can have different forms. They look different. So the first one is a volumetric image. The next 2 are surface-based images. So that's one thing. Then the second observation we can make is that for the third study, the S120, you can see that with the FSL software, there is no image. And there is no image for a good reason, because the type of analysis that was done in there was not actually achievable with the FSL. And so just now, I'm not going to detail the neuroimaging results too much, but I'm going to focus just on the first study. So if you look at DSO1, which was the first study we produced, first you can see that on the bright side, the activation, so in yellow to red, you have activation levels, and in blue to white, you have deactivation levels. So regions whose activity decreases when the task is performed. So first, you can see that the general pattern could be reproduced from the original paper. The second high-level observation we can make is that probably the FSL software is closer to what we had in the original paper. And this was good news to us because actually FSL was the software used in that paper. But of course, here we're looking at brains by looking at just a single image that represents a full 3D volume. So if we look more closely, and what you have here is multiple slices that represent the very same results, you can see even more differences than what we could see with just one slice. We have some clusters that get bigger, some small clusters that might disappear, and so on and so forth. And here we cannot compare with the original paper because all we have in the original paper are the images in the PDF. We don't have the images shared that would let us explore the full 3D volume. And then we've done a number of quantitative metrics, including dice coefficients, so that we could compare those activation maps throughout the brain. Then as a second step, what we've done is to go one step earlier. So we know that the last step that is performed in an fMRI pipeline is to threshold the statistic map to get what's significant and what's not significant. And we know that thresholding effect can lead to a lot of differences because basically a small change in the original map could have strong effect in the thresholded map if the value change was close to the threshold. So here we looked at the unthresholded maps before the thresholding. And here we can see probably that the patterns are more similar across software package than what we could see with the thresholded maps. And this was also seen in the correlation where we can see that probably the masks are, even though it's hard to compare correlations to dice metric, but that makes sense and that's consistent with the idea that basically differences accumulate throughout the pipeline. So the conclusion of that study was that the choice of neuroimaging software had an impact both on the final activation map and on the statistic map, but probably to a smaller extent on the statistic map. And of course, as soon as we did this experiment, the feedback from the community was, okay, but can you trace back, understand what was making results across neuroimaging software different? And so in another follow-up study, what we've done here, so here we're looking at the results of the first reproduction, if you remember, that was called DSO1. And basically, we looked at, we identify with our neuroimaging expertise, 5 different aspects within the pipeline that varied in the software. But we have to remember that here in the previous study, what we did is align the pipeline as much as possible with what was made available to practitioners. So the remaining 5 here corresponds basically to built-in options in the software that are not accessible to the end user. And basically what we've done is that we've done hybrid pipelines where we go all the way. So here you can see the blue line is completely under AFNI, means that it's a complete AFNI pipeline. And then 4, the blue line is all under FSL, it's a complete FSL pipeline. And in between, we are mix and matching. And of course, we removed the number of steps here, but the idea was to find a step where we'd have a big difference in the image, even though we were varying only 1 step. And so for this particular study, we could see that changing the subject level signal from AFNI to FSL had a strong impact on the level of activation. The good news is that we were able to find one main contributing factor for each study. The not so great news is that it was different for each study. We think that this is important because if you go back to domain experts, if you are able to say, okay, if you say there are differences across software packages, it might be difficult to understand what's going on. If you go back to domain expert and say, okay, when I change the way the subject level signal is represented, it creates differences, then we can starting to make hypotheses and understand what's really happening. Okay, so I've illustrated how reproducibility in the case of using different pipeline can lead to different results with two different experiments. One where basically we had different teams of experts all worked with the same data set and trying to answer the same research questions. And the second study where instead we fixed the analysis, but we only changed the factor that was the neuroimaging software used. But of course, those results, when I share those results, typically what the community is requesting is to find solutions and deal, now that people understand that analytical variability exists, how can we deal with it in an efficient manner? And so for this, I'm going to finish by presenting two projects that are more like open science projects where people can take part and help investigating analytical variability. And I hope that as a community, we can find proper solutions. So the first one was done by Elodie Germany in collaboration with Elisa Fromont. And here, basically, what we've done is that we worked with the HCP, so Human Connectome Project Young Adult Dataset. That's an open dataset in neuroimaging that is extremely large in terms of the number of subjects. So we have over 1,000 participants, which is really, which is quite big for our community. And what we've done is that we analyze the same data using different pipelines. So two software packages, different parameter for pre-processing and different parameter for the statistical analysis. Those parameters do not explore the full spectrum of possible different pipelines, but they were used as parameters that we often see varied in the literature. We worked with 5 motor tasks, so moving right hand, left hand, right foot, left foot, and the tongue. And we've built 1,000 group of 50 participants. And the idea was to try and study the variability observed in the results when we have different analysis. An important point here that is not limited to neuroimaging, but that is quite typical in biomedical research, is that we have no ground truth. So we can't just say, okay, this pipeline gives the results that are closest to the ground truth, I'm going to keep this one. Typically, we have no. So what we've done here is that basically we tried to cluster the results from different pipelines. And here's an example from right hand. And basically we identify 4 different clusters. And those clusters could reproduce quite well across participants. So that's good news because basically we're building these maps for the different participants and different group of participants. But they were different across fMRI tasks. So basically, when you're moving your foot and moving your head would not lead to the same clusters of pipeline. So that was the first effort to try and understand the analytical space and reduce the complexity a little bit. So I said I was hoping this could become a community effort because basically as a result of this study, we shared the data set. So basically all the 24 reanalysis with the 1,000 group 5 motor tasks are shared in a data set that is called HCP multi-pipeline that is available on the public neuro repository, which is an GDPR compatible repository. So I would encourage people interested to go and dig with this data set. And then the second effort, so I've already mentioned NARPS, the mini analysis project. So this is a follow-up to NARPS that is called NARPS Open Pipeline, which is led currently by Boris Clénéo, was initiated by Elodie Germany and got contribution from Jérémy Lefort-Bénard. And so basically when the NARPS investigators recruited the 70 teams, they asked them to share their results. And for their results, what they requested was a yes or no answer, obviously, plus a confidence level. So people could say, okay, I'm very confident, not very confident. brain images. So we have the thresholded and unthresholded maps we looked at earlier. And then the pipeline itself was described using a textual description, but textual that had to fit with community guidelines that are called the COBIDAS guidelines. But so that's one way to make the description a bit more harmonized across teams. Nevertheless, COBIDAS leaves a lot of space for people to give less or more details. And in practice, when we looked at the different traces given by the different teams, the lengths vary a lot, and there are a lot of differences, and it's not that easy to compare pipelines. This, of course, was shared for the 9 research questions, but there was no code requested. I'm not blaming at all the investigators. It's still not common practice in our community to share code, and probably would they have requested code, they would have had much less analysis teams. A number of teams did share their code, but in general, the code was not shared. And so the goal of NAPS Open is to basically from the pipeline description in COBIDAS to rebuild the code so that we have a more formal representation of the pipeline that was applied, and that we can start thinking about how close they are, how different they are, how we can mix and match and get a better understanding of pipeline variability in the scientific community. So NAPS Open Pipeline is a collaborative project. We've been advertising it and working with contributors in different hackathons, and it's publicly available on GitHub, and of course, welcoming contributions. And to finish, I'd just like to give a little bit of a vision of what would be my dream if we can solve that issue together. So of course, that's a bit of an exaggeration, but we can say that the status quo right now in brain imaging, but probably in many other fields, is that we are going to present our results with respect to a unique analysis. Of course, that's a bit of an exaggeration because in a lot of paper, you might have a few variations so that people actually test how robust their results are. But this is mainly true. So my dream is that in the future, we have a way not to investigate the full space, because of course that would be computationally very expensive, but that we could guide practitioners in the exploration of that space. So maybe we could say, okay, that was the original pipeline, this is your problem. As a methodologist, I can tell you, you can try this 4 pipeline, and then that's going to be a stability check. You check that your results remain robust. And then you can test these other 4 pipelines, and those are the main axis of variability. And then as an expert, you can interpret what you see in the results and the differences you see in the pipeline, and explain how this relates to your research problem. So we have a number of ongoing projects that relate to this, including a better modeling of the analytical space. So right now, it's still difficult to understand which part of that really big space of pipeline is actually used in the community. We have effort to trace provenance because this is a good way to have a window into what people are using. And we have efforts building on software engineering study of different configuration to actually explore the analytical space at large. And with that, I'm going to thank you very much and end my presentation.